pre-incubated with astaxanthin and then exposed to singlet oxygen for 10 minutes and left in culture for 24 hours. Protection was measured by level of cell viability rate, cell survival, or cell resilience, as we talked before. In the astaxanthin-treated sample, cell viability was 95%. On the right, there are 3 images. They are very self-explanatory. In the singlet oxygen exposure, fibroblasts were highly destroyed, while in the singlet oxygen exposure with astaxanthin, the fibroblasts were highly protected. This experiment compared human fibroblasts were incubated with astaxanthin and then exposed to singlet oxygen for 24 hours. The superiority of astaxanthin is outstanding. There cannot even is a number 2 in these slides. Very difficult to find out a number 2 as the strength is so powerful against singlet oxygen. Now, let's find another very powerful study show how effectively astaxanthin, cantaxanthin, and beta-carotene protect our endogenous antioxidant protection, namely catalase and superoxide dismutase after 24 hours exposure to UVA irradiation. So in the first graph, astaxanthin keep our catalase resources stable, whereas beta-carotene and cantaxanthin see a significant deplete of our antioxidant resources. Now we are a little bit experiencing other problem here. So, okay, yes. The same happens in the SOD activities. Astaxanthin protect and actually increase SOD resources by 4%, while cantaxanthin and beta-carotene deplete SOD by 10 and 6% respectively. Fortunately, we have some problem. We cannot here. Okay. So astaxanthin, this slide, what tell us? Astaxanthin synergize with our endogenous antioxidant system to protect skin from premature sign of aging. Now, astaxanthin, we saw it before, has also powerful application to other parts, biological activity, including eyes. Actually, over 10 clinical studies suggest that astaxanthin provide antioxidant protection in the aqueous humor, in this zone. Inhibit inflammation in most part of the eyes, improve capillary blood flow in the retina, and improve resilience of the ciliary muscles. Now, I want to pay attention particularly to the ciliary muscle, where astaxanthin is particularly effective. This ciliary muscle control the lens thickness in order to focus the light on the retina. In heavily visual workload, the eye is focused on a fixed object distance for extended periods. That will cause muscle pains and fatigue. Clinical studies suggest that astaxanthin enhance accommodative refraction and accommodative contraction, refraction and contraction. That is very important part. Indeed, there is powerful clinical study where astaxanthin, this is a randomized placebo study, show the astaxanthin enhance accommodation contraction time with 46 fatigue subjects. The study examined various doses. High rate of accommodation function measure indicate the speed at which the ciliary body react to the direction of focus. The study reported a very strong improvement. 4 milligrams of astaxanthin daily for 28 days improved by 19%, while with 12 milligrams saw an improvement of 65%. Very impressive. Now, in this other randomized double-blind study, 40 eye fatigue subjects took 6 milligrams of astaxanthin or placebo for 4 weeks. So after 2 weeks, the astaxanthin group reduced accommodation rate by 36% and 39% after 4 weeks, compared to almost no change in the placebo group. The study is also relevant because it shows the efficacy of astaxanthin on population over 40s. Now, we talked now about the eyes, we talked about the skin, we talked a little bit about endurance. Now, the new emerging studies are very pushing on astaxanthin research on cardiovascular. Recent study shows that astaxanthin reduce the 10 factors that lead to atherosclerosis or lead to development and aggravation of atherosclerosis. Before delving into the details of the study, the picture gives an overview of astaxanthin effects. The fireman, starting from early stages, astaxanthin increase HDL cholesterol, decrease lipid peroxidation, and reduce triglycerides. In the developing stages, astaxanthin decrease macrophage inflammation, reduce plaque formation, improve vascular tone, reduce blood pressure, and increase plaque stability. In the advanced stages, recent studies show that astaxanthin reduce plaque rupture and improve blood flow. I will show now some study because otherwise we look like just a panacea that does everything. But show me the meat, show me the studies, and that's what I'm going to do now. Pass on that. So, in these slides, macrophage infiltration, so macrophage that lead to inflammation in the endothelium especially, and in this case in the atheroma, is closely associated with plaque stability, and therefore potential reduction of plaque ruptures. Astaxanthin significantly decrease macrophage occupied lesion areas in the thoracic aorta of rabbits. To be more specific, astaxanthin decreased macrophage infiltration in the atheroma by 40%. Let me repeat. Decrease macrophage infiltration in the atheroma by 40%. Now, there is a description here. It's not easy to read these graphs, but we can see this is the atheroma pictures, rich in apoptotic cells, which is mostly the red ones, and macrophage, which is brown. Now, open arrows indicates apoptotic macrophages, and negative macrophages are marked by arrowheads. Now, it's not easy to explore, but we can see that from astaxanthin, it decreases the macrophage infiltrations. Maybe we can see in the studies much better analyzed in more details. But in this slide, I would like to show how astaxanthin, so this is the pictures in numbers, how astaxanthin decrease apoptotic cells and necrotic areas in the rabbit aorta. 4 milligrams of real astaxanthin for 24 weeks decrease apoptotic cells by 42% and necrotic areas by 17%. Now, this slide is simple as powerful. Rabbits, actually hyperlipidemic rabbits, that ingested 4 milligrams of astaxanthin for 24 weeks decrease rupture plaques in the arteries by 49%. I would say 50, but 49% to be specific, compared to control group. Whoa, that's big. Now, I prefer pictures. I believe in pictures. So the slide introduced the effect of astaxanthin on arterial wall thickness, width, and structural features. So hypertensive rats ingested 5 milligrams of astaxanthin for 7 weeks. Now, compared to control group, this is the astaxanthin group on the right, compared to control group, which actually is olive oil. So I don't like to show this study, being from Italy and big fans of olive oil. The astaxanthin reduced the thickening and increased widening of the coronary arteries. Astaxanthin also improved the vascular tone, elastin band in the aorta by 36% in spontaneously hypertensive rats. You can see quite a difference. And in the third picture, we can see the astaxanthin ameliorated structural changes in the blood vessel. Fewer and straighter elastin features compared to the control groups, which show intense and branch elastin features. Now, how the story turns, because at the end of the story, it's resilience against degenerative diseases. So this is the end of the story. This is a very fascinating experiment that involves hypertensive rats. These hypertensive rats, they become stroke prone at later stage of their lifespan. In this experiment, the rats ingested 50 milligrams of astaxanthin for 10 weeks. Afterwards, the treatment was suspended. The incidence of stroke reduced by 50% to control group, and I mean 50%. Sorry, I get too excited when I see these studies. So now astaxanthin, just very quickly, we know that, for example, in Canada, over 7 million people suffer from metabolic syndrome. And several studies have shown that astaxanthin has several beneficial effects. Today, I quickly introduce two recent studies that show impressive potential of astaxanthin and lipid metabolism. You can see it in this study. I don't know why here something is going on with the writing, but this clinical study showed the powerful anti-lipid peroxidation power of astaxanthin in the blood. 20 healthy non-smoking male ingested 8 milligrams of astaxanthin every day for 3 months. Subsequent analysis show that a sharp reduction of lipid peroxidation, actually as sharp as 60% and 36% in plasma 15 fatty acids and plasma 12 fatty acids. This is the last study. The slides show that astaxanthin decrease triglycerides and increase HDL good cholesterol. Now, this is a recent double-blind placebo study with 61 moderately hypertriglyceridemic volunteers. We ingested 6 milligrams, 12 milligrams, and 18 milligrams of astaxanthin supplementation every day for 3 months. So, triglyceride decreased by 25%, 12 milligrams, up to 25%, and good cholesterol HDL increased up to 15%. That is very, very powerful. Now, where astaxanthin, real astaxanthin come from? Well, to obtain astaxanthin from Haematococcus pluvialis, Fuji cultivates the algae in the photobioreactor here located in Gothenburg, Sweden. Sorry about my pronunciation. Established in 1987, Bioreal Sweden is the first company in the world to commercially cultivate Haematococcus pluvialis. This closed system is the safest and indeed the most effective to extract astaxanthin. Astaxanthin has to be extracted during its high peak concentration in a coincidence with the lowest chlorophyll concentration. This is a crucial procedure because chlorophyll derivatives, also called phospholipid sometimes, can cause toxic effects. After being extracted and produced in Sweden, the final production is in Japan, in Toyama, where there is the Fuji headquarters, and where the Haematococcus pluvialis is extracted and purified. Then the astaxanthin raw material is checked for quality and released for sales worldwide. Dr. Perricone, I